So this next section of this chapter talks about genetic changes. And genetic changes are alterations in the nitrogen base sequence of DNA. They can be good or bad, but not all changes in DNA automatically result in alterations or changes in amino acids. So if you recall from the last chapter, let's go back to the last chapter, some of the chapter text. It turns out that codons in DNA represent ultimately codons in messenger RNA. If you want to get technical, there really are no codons in DNA. There's codons in mRNA, but DNA sequences represent codons in mRNA, which represent amino acids. But it turns out there's redundancy in the code. You may remember this from the table here. The GGG and GGA and GGC and GGU all represent the same amino acid, glycine. So if there's a change, if a DNA sequence CCA changes to CCG, then the GGU or the GGC or whatever codon is a new codon is being transcribed, it won't affect the protein because it's still glycine that's going to be placed in the protein. But if the first or second nitrogen base is changed in a DNA sequence or an RNA codon, well, now that is probably going to change the amino acid that's represented, and as a result, there's going to be probably either a good or bad effect on the protein product. So that brings us back now to this chapter. Let's talk about the mutations having positive or negative effect. So DNA mutations are generally not neutral unless the amino acid was not changed. If a DNA mutation is positive, that means it enhanced the fitness level of the living thing that the mutation is a part of. And if the mutation's negative, it actually decreased the fitness level. The thing is, fitness level depends on environment as well as it depends on metabolism. So a good case in point is malaria. Malaria is caused by a single amino acid substitution at the number 6 position. Hemoglobin A is normal hemoglobin that's found in red blood cells. Hemoglobin, if you don't know it, it consists of 2 hemoglobin A units and 2 hemoglobin B units. Alpha and beta is what they're called. And each one can carry oxygen. So 4 polypeptides actually make up a single hemoglobin. Well, if the hemoglobin A units in the number 6 amino acid position have glutamic acid, they're normal hemoglobin A. And if they have valine at the number 6 amino acid position, they're actually hemoglobin S. So people who carry sickle cell anemia, half of their hemoglobin is hemoglobin with hemoglobin S in it, and the other half of the hemoglobin is this type with just normal A. If a person actually has sickle cell anemia, it means all of their hemoglobin is of this type, and that actually is a major health problem. So how do we determine the effect? Is it the positive or negative effect of a mutation like sickle cell anemia? Well, you have to consider the evolutionary history and the environmental effect of the mutation. Turns out that in parts of the world where this parasite lives inside of mosquitoes, Plasmodium falciparum is the scientific name of the most vicious form of malaria. It's the most aggressive, the most debilitating, and most fatal. So maybe 20% of all people who get infected with Plasmodium falciparum will die without medical intervention. The people that get infected with Plasmodium falciparum that only have this kind of hemoglobin have at least a 1 in 5 chance of dying. If people are carrying some of this hemoglobin in their bodies, the Plasmodium falciparum cannot kill them. They're immune to the malaria. So as it turns out, if you have sickle cell anemia and you live here in the United States, your mutation is negative because we don't have Plasmodium falciparum here, and our Native American Plasmodium parasite was Plasmodium vivax. It's rare in the United States now because of mosquito control. So in Africa now and in parts of Asia where Plasmodium falciparum is found, if you have a sickle cell anemia mutation, if you're a carrier, the mutation actually increases your fitness, and the mutation is having a positive effect on you. So as it turns out, the sickle cell mutation actually causes abnormal hemoglobin polymerization in red blood cells, especially if oxygen levels get low. So in this picture right here at number 1, that boomerang-shaped cell is actually a red blood cell, which should have been perfectly round like that, and instead it's shaped like a sickle, like that. Those cells, the ones that are sickle-shaped, they can clot your blood and cause spontaneous strokes and tissue death in your body. So if you are homozygous, meaning if both of your hemoglobin A genes are of the hemoglobin S type, in other words, if you cannot make hemoglobin A, you're going to have to lead a very sedentary life, because if you get up and run or exercise or anything, your cells will sickle and your organs will be deprived of blood as your blood clots. But if your genotype is hemoglobin A, hemoglobin S, in other words, if one of your genes is the normal A type and the other gene is the S type, well, now you actually have almost no chance of the blood clotting problem, but you're immune to malaria. But this genotype is still at least slightly negative if you don't live with Plasmodium falciparum. If you do live with Plasmodium falciparum, this genotype has a positive influence on your fitness. So mutations are positive or negative, but to determine the effect, you have to take a look at what the mutation is doing in the environment the organism lives. Now, how do mutations happen? Well, one of the ways, and we're not going to go into it too much, but one of the ways is radiation. Radiation exposure will literally fragment chromosomes and cause spontaneous mutations. Other chemical insults to your chromosomes, there are lots of chemicals. Many of them are unnatural ones that are man-made. Stuff like chlordane and pentachlorophenol and endrin and dieldrin. Those are pesticides that were actually manufactured back in the 1950s, widely used in the 60s. We now know that they caused all kinds of DNA problems in not only humans, but all kinds of life forms, and that's why they're all banned. Those chemicals actually slip into the double helix and bind to our nitrogen bases and then interfere with normal operations of replication, which results in changes to the DNA when DNA gets replicated. So that's why those chemicals have been banned. Now, outside of that, and outside of chemistry and radiation, mitosis sometimes and meiosis mess up. And they actually don't just change genes, they change the numbers of chromosomes you have. In animals, usually this kills the embryo, but in plants, it often results in more healthy and vigorous growing offspring. So strawberries, for example, are pentaploid, and they're all cloned. So the wild strawberries are very small. They're about the size maybe of the flesh under your pinky fingernail, and those are diploid strawberries. But pentaploid strawberries are gigantic and look like tumors. And those are the strawberries we grow. The only thing is they're pentaploid, and so those strawberry plants can't successfully make seeds, so we have to clone them, which is a problem when you're growing them in big fields where bugs are crawling around and trying to eat them. But lots of our agricultural plants are polyploid, meaning they have more than the 2 sets of chromosomes than they should. Generally speaking, without human intervention, polyploid plants often fail in the long run because they often can't sexually reproduce. But with human cultivation and human care, those plants can be kept alive despite the fact that they're clones and parasites are trying to destroy them, um, they're saved by people pulling weeds and spraying herbicides. So, banana plants are triploid. They're very vigorous growing and make huge fruit. Wheat and corn are both polyploid plants and are made by a really special hybridization process. They are grown from seed, but often the seeds themselves then, when they grow up and make big tall plants, the plants themselves don't reproduce well. So errors in chromosome number are usually very bad for animal embryos. Errors in chromosome number in plants are in the long run bad, but in the short run, they do make some vigorous growing offspring. There's a few chromosome anomalies in humans that are tolerable. Trisomy 21 leads to Down syndrome, and that causes the mental or a brain effect. They usually have a very kind of a happy disposition. Down syndrome kids tend to be short of stature. They have a rounded face. Those kids have 47 chromosomes in every cell, and they have a triple copy of chromosome number 21. So that's why it's called trisomy 21. Trisomies of most of the other human chromosomes are fatal. So trisomy 1, trisomy 2, trisomy 3, they have never been found walking on Earth, but they have been found in the cells of miscarried embryos. Klinefelter syndrome is another one of the few chromosome anomalies that a human can survive. In Klinefelters, you have 3 sex chromosomes, 2 X's and a Y. These individuals are born boys. They're males at birth because they have the Y chromosome. But at puberty, they get a double secondary sexual characteristic maturation. They get breast formation, hips widening, body fat deposition, as well as facial hair and some muscle development and a deepening voice and an enlarged Adam's apple. So they call that pseudohermaphrodism, or some people just call it hermaphrodism, but those folks actually don't have a penis and a vagina. They have normal male primary characteristics, but they get female secondary characteristics. So that is one of the chromosome anomalies that's survivable with humans. Another one is Turner. Turner is where a baby is born 45XO. So just to list this out, trisomy 21 is 47XX, that would be a girl, and then T21, trisomy 21, or 47XY, trisomy 21. Those would be girls and boys with Down syndrome. Klinefelter syndrome is 47XXY. Now, those individuals, like I said, they're born boys, and they are a mix at puberty. Turner syndrome is 45XO. The first number, I should say, represents the number of chromosomes you have, and the second numbers represent your sex chromosomes. That's how you actually represent a karyotype. This is the Turner karyotype right here. This is a picture of the chromosomes taken from a baby that had Turner syndrome. You'll notice here, every chromosome there's a pair. Here's chromosome pair number 1, pair number 2, pair number 3, pair number 4, pair number 5, and so on. Sex chromosomes, we got 1, we got the X, we don't have the Y. So this individual is called a 45XO. There's 45 chromosomes in there. Now, the karyotype on the right is pairs of chromosomes, and then when you get down here to the bottom, there's 2 X's and there's 1 Y. So this is 47XXY. This is Klinefelter syndrome. Normal karyotypes are 46XX or 46XY. That's 46XX is girls and 47XY is boys. To make things more complicated, it turns out that some babies with Turner and Down syndrome, they're mosaics. What a mosaic is a person who has different cells in their body with different chromosome or genetic constitutions, different genomes. And if you're a mosaic, that means some of your cells if you were a Down syndrome kid with a mosaic pattern, Some of your cells are 46XX, and then some of your cells are 47XXT21. So that means that one of the early mitotic divisions, when you were an embryo, there was a mistake. One cell was shorted a chromosome completely. The other cell got the extra. The one that was shorted died. The one with the extra lived and went on to affect the body, and the individual has Down syndrome issues. So the bottom line is these problems here are all animal problems caused by meiosis or mitosis problems, okay? Whereas these problems up here are issues with plants. Polyploidy in plants is a plant issue only. No human babies have ever been born with polyploidy. Embryos have been documented with karyotypes that were triploid. In other words, and we think that's 2 sperm hit 1 egg, but they do not survive. Embryonic development is too complicated and they don't survive. So now we need to move on. There's other ways that your genes can have issues. And it turns out that genetic changes can actually make things much better for you. So changes in a genotype is going to affect your phenotype probably, and then natural selection is going to get a hold of that. So for bugs living on plants, if you have a situation where you have a bunch of bugs eating on like corn plants in a cornfield, there's going to be some genetic variety in the cornfield, and you're going to be a few freakazoid bugs that are just kind of weird, and they're not like the other bugs. If you spray herbicides on the field, you will end up wiping out all of the bugs that are vulnerable to the herbicide. And you end up, with a whole bunch of the bugs that like here are blue, they're all killed. But this little goofy red bug, it's got some random resistance to your herbicide, so it doesn't die. As a result, this bug, through many generations of growth, becomes the most populous bug, and this bug then becomes rare. That's exactly what happens with pesticide resistance in fields. You first kill all of the vulnerable bugs, and you leave behind the genetic variants that are not vulnerable to your herbicide. We've already talked about sickle cell anemia and how that works, and it's true for every genetic trait. With humans, we cheat a little bit. We use antibiotics to keep ourselves from dealing with fitness problems too much. But in general, all other organisms are constantly dealing with environmental pressures. And all they've got in terms of the card game is chromosomes to produce proteins that either will help them or will not help them survive and have babies in their environment. So now we got to talk briefly about, okay, so what are other ways that DNA changes? So in bacteria, there are all kinds of ways that DNA in the genomic part of the bacterial DNA that they have. There's 2 types of DNA in a bacteria. There's plasmid DNA and genomic DNA. And we're talking here mostly about genomic DNA, but also plasmid. You'll see here in a second. Bacterial transformation. That was discovered by Fred Griffith. Basically, that's where bacteria just absorb naked DNA. So if a bacteria bumps into a naked piece of DNA just floating around in solution, it's sort of like somebody walking down the hall and seeing a quarter on the floor. You just pick it up. It's not yours, but now you own it. And bacteria are fond of doing that with DNA. And what they'll do is they'll transcribe and translate the protein that's on that DNA they picked up, and it might give them an advantage. And as a result, then they survive better. It's just like the quarter allows you to go buy something and have something for yourself, some candy or something that you otherwise wouldn't have gotten. Transduction is the acquisition of viral DNA in a bacteria due by attack by a virus that likes to shoot its DNA into the bacterial genome. So transduction is really common in bacteria. It turns out that tuberculosis bacteria are actually harmless until they are transduced by a virus that turns them into lethal killers. It turns them into a human superparasite. That is a case of what we call DNA transfer by transduction. So that's another way. So transformation and transduction are 2 different ways that bacteria willfully pick up new DNA either through their interaction with a virus or their interaction with DNA just floating around in solution. In the case of the virus, the bacteria probably really would rather not have the virus shooting its DNA in it, but sometimes it helps. And then there's other ways. Conjugation is where two bacteria actually touch each other and they trade plasmids. It's similar to transformation, except in conjugation, two bacteria actually are exchanging bacteria with each other, whereas in transformation, it's a bacteria just sucking up a piece of DNA that's floating around. Conjugation is extremely common and a major problem in medicine, where bacteria of different species trade antibiotic resistance plasmids and cause problems in hospitals. Never a good idea to walk around in a hospital, touch things, and then touch your face, because you're putting conjugation changed bacteria on your skin surface, which then can lead to huge problems. The last type of DNA movement system in bacteria that is not directly involved in bacterial reproduction is called transposition, where DNA in a bacteria moves from one part of the genome to another. That was first discovered by Barbara McClintock way back in, I believe, the 50s. She didn't get the Nobel Prize, though, until 30 years later when science caught up with her amazing discovery. She discovered transposition actually in corn. As we now know, human DNA transposes, but so does DNA inside of a bacteria. Transposition of DNA means that pieces of DNA will move, say, from here over to here. And the reasons for that are unclear in many cases, but it happens. So that can result, if transposition does not happen in a proper fashion, you're going to get changes in the DNA, and then you're going to get changes maybe to protein production. Now, eukaryotes have a number of ways to try to ensure genetic variety so that we're all not the same. Being the same is not a good idea in the long run, because natural selection will catch up with you as parasites discover the Achilles heel that everybody has in their genome. So it turns out that crossing over is a big deal in meiosis. Crossing over allows maternal and paternal chromosomes in an organism to exchange DNA. And by doing that, you're changing the gene linkage patterns in chromosomes through the generations, ensuring that as parasites are co-evolving with organisms, they can't gain the upper hand. Secondly, random chromosome assortment during meiosis. When the maternal and the paternal homologs separate, if this is the M for maternal homolog and this is the paternal homolog, and the spindle fibers are pulling them apart in the cell, this is the cell. If the maternal homolog is on the left, it'll end up going to the left, and if the paternal homolog is on the right, it'll go to the right. Humans have 46 chromosomes. They line up in 23 pairs. You could theoretically get eggs and sperm that have all maternal DNA or all paternal DNA. That's really rare, but it could happen. Usually you get a mixture. The actual content of maternal and paternal DNA is random because of the way the chromosomes line up and the way that crossing over happened between the homologs. Okay? And then lastly, fertilization. Having to go out and find a mate that is not your brother or sister also makes sure that your kids will be different than you were. So animals and plants and other complex animals like fungi gain an advantage of genetic variety by using these systems, crossing over, random chromosome assortment, and fertilization to make sure that their offspring will live long and prosper, because they won't be the same as what they are. Now that brings us to viruses. Viruses have really strange ways of reproducing. First off, they're not technically even alive, but because they reproduce in eukaryotic cells, they actually disrupt eukaryotic cells, and as they do that, they can change the eukaryotic cell genome as well as their own genome. So viral replication it differs from other reproductive strategies, and it generates genetic variation in a variety of ways. Viruses are designed for rapid reproduction. Basically, the reproductive cycle of a virus helps to make sure that genetic information gets transferred from them to their, you know, their offspring, if you want to call it, viruses. But in so doing, they often mix their genes up with their cellular hosts, and that can create interesting, situations in terms of genetic variety. The typical viral life cycle involves the virus attaching to a cell like this. The virus injects its nucleic acid, and then the nucleic acid often becomes part of the genome of the host. Okay, this is called right here. This fusion of the virus into the host is the virus lysogenizing its host. Now, many viruses don't actually do this. They inject their DNA and then they jump right to this. The ribosomes in the host cell manufacture more viral proteins. Enzymes in the nucleus of the host cell make more viral nucleic acid, and then the virus jumps right to assembly of new viruses, the destruction of the cell, and the infection of new cells. But the viruses that actually fuse their genome into the host, they become part of the host. Viruses like HPV, the human papillomavirus, they tamper with the host cell genome. In so doing, in humans, that dramatically increases the chance of cervical cancer in females and probably prostate cancer in males. That's a problem. The typical viral life cycle either then is lytic, meaning not involving host cell genome tampering, or lysogenic, involving host cell genome tampering, or it's both. In the case of AIDS viruses or HIV, those viruses actually, scroll a little bit here, those viruses actually do both. When you initially get infected with HIV, they actually fuse into your T cell genomes, and as your T cells reproduce, you get more and more T cells with HIV viruses in the genome, and then they switch to a lytic cycle. The viruses destroy your T cells as they come out and infect other T cells. So lysogenic viruses tend to do both of these. Lytic viruses, on the other hand, tend to just do this, and they skip this DNA fusion part. So basically, viruses do not have nucleic acid repair systems, which means that they're more prone to genetic mistakes, either by just random radiation or chemical insult or accidental replication issues in host cells. They have no way of fixing those mistakes because they're at the mercy of the host cells. As a result, the viruses are already prone to more rapid evolutionary change than living cells. And then, and this is crazy, but this is how influenza creates crazy strains. If two related but different strains of viruses, like a bird flu virus and a human flu virus, infect the same host cell, you'll end up, so if two different viruses inject their material into the same host cell, you'll get genetic recombination of the different viral particles, and the viruses that escape will be new. They will be brand new types of viruses that have never been seen before. Many biologists think that was part of the story of the 1918 strains of influenza that killed maybe 60 million people worldwide. The viruses were different because they were different strains of influenza brought into a single viral container. So that's strange. And then lastly, HIV resistance is always a problem for people taking antiretroviral drugs. And if a person is taking the drugs and they just take off for just a few days and they stop taking the viral drugs, the antiviral drugs, they can end up with a whole new strain of virus a week down the road that really was rare in their body before or wasn't even detectable. And then now suddenly they have a whole lot of that virus in their body. So the antiviral drugs keep DNA replication to a minimum. If you go off the drugs for a little while, you'll get massive viral replication, and you'll have viruses that are now different because of just random, ridiculous mutations that occurred because of the way viruses reproduce. And as a result, when you go back on the drugs, you'll stop all the vulnerable viruses from reproducing, but the resistant ones now can get a foothold. So that kind of concludes this portion of the chapter that talks about the ways that DNA actually changes.